I'm very excited about the talks today. I'm very excited also to welcome Dr. Lauren Gillies Campbell and Dr. Olivia Mason to talk about some of their research. First, we'll have Dr. Gillies Campbell, and then followed by that, we'll have Dr. Mason for the second part. And though the kind of theme or title today of the presentations are oxygen as a driver of microbial community structure and diversity in the northern Benguela upwelling system. So Dr. Gillies Campbell was a 2017 RGNO participant. Lauren earned her PhD at Florida State University with a PhD in oceanography, and her research was focused primarily on studying microbial community in the Gulf of Mexico dead zone. After graduating, Lauren worked in the lab at Florida Department of Environmental Protection, where she did microbial source tracking to protect the health of people in the environment in Florida. Now Lauren is a Basin Management Action Plan Coordinator for the Florida Outstanding Springs and Spring Sheds. Lauren works with Florida stakeholders from local governments and agriculture to identify sources of nutrient loading and reduce those loads of groundwater in order to protect Florida's valuable springs and water resources. Welcome, Lauren, and for any questions, you're welcome to type them in the chat, and then when we have a break, then I'll call on people to ask their questions live, or I can read the chat for you. Lauren?
Great. Thank you so much, Jake, for that introduction. And it's wonderful to see you all today. So I'm really happy to be here and talk with you all a little bit about the data set we collected in 2017. So like Jake said, I was a 2017 participant of the RGNO course while I was a graduate student at Florida State University. And what I focused on was looking at microbial community structure across oxygen gradients in the Gulf of Mexico dead zone. And so I was curious about other low oxygen environments, coastal low oxygen environments. And so today I'm going to talk to you about how oxygen is a driver of microbial community structure in the northern Benguela upwelling system. And here are my keywords. And I'm going to go a little bit into the background of the keywords, which I think will help throughout the presentation. So 16S RNA gene, it codes for one part of the ribosomal RNA that is present in most cells. So it's universally conserved. It has the same function in all cells, and therefore it's a really great tool to use to look at the microbial community. And we do that using iTag sequencing, which is an Illumina-based tagged barcode amplicon sequencing, and it targets that 16S RNA gene from environmental samples. We're going to be looking at oxygen minimum zones, and we have our data sets broken up into, we kind of characterized it by oxic samples, dysoxic samples, and suboxic samples to look at how the community changes across these oxygen concentrations in the water column. So all of these samples that I'll be talking about today are from the water column. I'll be talking about hypoxia, since that's where my graduate research was looking at the hypoxic zone in the Gulf of Mexico. And so when I refer to hypoxia, I'm referring to dissolved oxygen concentrations of less than 2 milligrams per liter, or 62.5 micromoles in the water column. And then I'm also going to be talking about the nitrogen cycle today, which is driven by complex microbial processes such as nitrogen fixation, assimilation, nitrification, anammox, and denitrification. So I'll be going into a little bit of the nitrogen cycle and what organisms are involved in that cycle. Specifically, I'll be talking about archaea, specifically Thaumarchaeota. So that's an organism that showed up to be really interesting in our data sets, and overall bacterial community, which refers to both the bacteria and archaea in our water samples. So I'll go a little bit into the Gulf of Mexico research, which really drove us to start looking into other OMZs. I'll talk about my methodology in the Gulf of Mexico, and then why we wanted to learn about the Benguela upwelling system, And then I'll go into the methodology I used in 2017 during the RGNO course and a little bit about what we found in those samples. And I'll be looking at the phylum level of the bacterial community. So we are really interested in looking at how the bacterial and archaeal community and diversity changed in response to dissolved oxygen across an oxygen gradient. And the goal of our research was to look at these linkages between the biology and the chemistry, specifically in the Gulf of Mexico hypoxic zone, to better understand their response to low oxygen conditions. And then to compare that microbial community to the microbial community in the northern Benguela upwelling system, because these are, while they're both low oxygen systems, they're different. The low oxygen is created by different physical factors and nutrient dynamics. So I'll go into the next slide. So the next slide describes what the Gulf of Mexico hypoxic zone is. So this is one of the second largest zones of human-caused coastal hypoxia in the world, and this is due to eutrophication. So here is the Mississippi River, and you have all this nutrient-rich water from all the farmlands out in this whole area, so this whole watershed that flow into the Gulf of Mexico. And this nutrient-rich water flows, creates phytoplankton blooms, and the phytoplankton uses the oxygen and further draws, so the phytoplankton degrade, and then the microbial community uses the oxygen from that degrading phytoplankton. So here, if you look at this figure, and then that draws down the oxygen in the lower, near the seafloor. And then in the summer months, it's really warm. So you have that stratification and not as much mixing in the summer months. So the dead zone only forms in the summertime in the Gulf of Mexico, which makes it an interesting environment to study. So I went out and collected samples to look at the microbial community in the low oxygen environment. And what I wanted from reading the literature, I wanted to focus on the nutrient cycling, specifically nitrogen. So I'm going to go into why we want to study these microorganisms and specifically their role in nutrient cycling. So microorganisms are really cool because they carry out necessary chemical reactions. So oxygen is the most favorable electron acceptor, but when oxygen levels decline, there's this diversion of energy to other electron acceptors. And they have this ability when oxygen declines to use these other tools to survive. So for example, we focused on the nitrogen cycle to look at how the microbial communities create these like niches based on these available electron acceptors as the oxygen decreases. So the sequence of redox reactions drives these niche differentiations, and these processes can be important to understand because they could potentially produce greenhouse gases such as N2O and contribute to the loss, as well as contribute to the loss of fixed nitrogen. So I'll be focusing on today of the ammonia oxidation, nitrite oxidation in the water column, because the hypoxic zone never reaches anoxia, so it's always a little bit of oxygen present in the water column. My methodology is very similar to what I carried out at RGNO. So I collected water column samples using the CTD, the Niskin bottles, and then used the CTD sensor to get oxygen. I also collected samples for chlorophyll and nutrients. And then I filtered seawater using a peristaltic pump and collected the biomass on a pre-filter and then a 0.22 micron Steravex filter. And that's what this filter looks like here. And then here I collected 5 to 10 liters of seawater. And I collected samples in 2013, 2014, and 2015. So we could look at how, based on how the hypoxic zone changes, because we saw that the hypoxic zone changed location, it changed size. So that really was interesting to look at over the years. And then back in the lab, we extracted the DNA. And the method we used in the lab was a phenylchloroform bead beating extraction method, and then we purified it. And then we got the quantity. And then we used the 16S RNA genes and amplified those and used gel electrophoresis to look at them here. And then we used the reverse primer, which contained a barcode, and were able to do ITAG sequencing. And that gave us, these sequences were then clustered into operational taxonomic units. And this is what I used for my 2013, 2014, and 2015 data, but for the Benguela upwelling, the methodology changed a little bit, which shows how fast the field evolves based on new and improved technologies. So we'll go a little bit into that slight change in methodology for the Benguela samples, since these samples are from, 2013 and 14 and 15. So here's a map of where I collected these samples, and you can see below here the areas of low oxygen are in blue and purple. So you have this area of hypoxia that forms off the Gulf of Mexico. And then here are the samples, and then this is where it is in reference to the Gulf. And important to note is that this is a shallow continental shelf where the average depth for the sites was around 18 meters, which is a little different than the Benguela upwelling system, which is I was able to collect a lot more samples from deeper sites there. So this is a shallow coastal environment. So briefly, just looking at what we found. So these are our most abundant taxa. And we have a oxygen concentration. So our lowest oxygen sites going up to our highest oxygen sites. And the first thing we noticed when we looked at this data is the stom archaeota and how their abundances increased with decreasing oxygen. And so we can start seeing that potentially oxygen can really be driving the microbial community. And we found, based on doing some diversity statistics, that there was a significant difference between oxic and hypoxic samples. So we saw similar organisms between all of our samples, but their abundances vary between oxic and hypoxic sites. So here's a map to really show you visually where the low oxygen area is and how this stom archaeota, we almost have this hot spot in this area of low oxygen. So it's around, deeper, a little bit offshore, but you can see it really correlates nicely. And so this OTU, the 16S RNA gene, was highly similar to an organism called Nitrosopumelus maritimus. And their numbers accounted for more than 40% of the microbial community in our hypoxic samples. I also did qPCR to get the 16S RNA genes to further back up our ITAG data. And we saw similar, higher abundances of the RNA gene in these low oxygen sites. So here I'm going to introduce the NMDS plot, which stands for non-metric multidimensional scaling, to show you different drivers of microbial community structure. So this is looking at our whole microbial community, and you can see how it clusters based on oxic and hypoxic sites. And then if you go over here, you can see our what we wanted to focus on. So our statistically significant environmental variables, as well as the organisms we are looking at. And you can see the stom archaeota, the qPCR data, and the OTU data correlates nicely with the low oxygen sites, so in blue, and then the oxic sites in red. And it was inversely correlated with dissolved oxygen. So now we're starting to really see how these primary environmental drivers and shaping the environmental community. And we see that the stom archaeota seems to be playing an important part in that, and they are a known ammonia oxidizer, which is really interesting. So we started Here's just my 2014 data. So I also wanted to show how over the years, we are still seeing this abundance. So this is 2014, and I collected surface samples that year. So we have from oxic to hypoxic, and you see in yellow, this Thamarchiota again is more abundant in our lower oxygen bottom samples. And here is comparing the 2 years together. And then this was interesting because in 2014, the low oxygen area was closer to shore and it was smaller. And you could still see that the nitrous cumulus OTU was closer to shore and their abundances were not as high as in 2013 because 2014 was a smaller hypoxic zone. So it's a really dynamic area that occurs every summer and therefore can influence in the environmental community and what they're doing nutrient-wise, nutrient cycling-wise in this environment. So why do we care? Nitrous cumulus is a known cumulotha autotroph, meaning that they can obtain energy through the oxidation of ammonia coupled with carbon fixation. So nitrification is a two-step process. You have the first step, and then the second step is carried out by nitrite oxidizing organisms. And so the Thamarchiota or nitrous cumulus can carry out this first step. So say if we have a higher abundance of this one organism, it could potentially lead to a decoupling and an imbalance in the nitrogen cycle in a low oxygen environment. And it has been shown that ammonia oxidizing archaea, or AOA, have the potential to produce the greenhouse gas nitrous oxide in the surface ocean and in low oxygen marine systems. And it has also been shown that nitrous cumulus maritimus have a high affinity for oxygen and ammonia. So usually it has been shown in the research that Thamarchiota increase with depth, but here and in most recent research, we can see that potentially it's oxygen that's driving their abundances. And they're also important because they're considered to be some of the primary nitrifiers in ocean ecosystems. So let's see what we discovered in the Benguela upwelling system, low oxygen environment. So we were interested in comparing the microbial community in this oxygen minimum zone to the one in the Gulf of Mexico. I wanted to broaden my dissertation story and look at a different coastal oxygen minimum environment. Now, while the Gulf of Mexico is eutrophically driven, so it has more of a human influence, it's been seen that oxygen minimum zones are expanding in our oceans due to climate change, warming waters, and this can really impact the microbial communities and therefore nutrient cycling in our oceans. When I went to the course, I spent a lot of time studying this map to the right to try to understand which sites I wanted to target and what depths I wanted to target. So here's a map of the site that they give you at the course, and then here are the sites that I collected samples from. And I went on the cruise that sampled the northern Benguela upwelling system. So that's why we're focusing on the northern part of the system. And I knew that I wanted to focus on the nutrient cycling and looking at low oxygen microorganisms. So I targeted the primary and secondary nitrite maximums, the chlorophyll max. And I was interested in the microbial community across the oxygen gradient. So I also targeted different oxygen concentrations throughout the water column. And my goal was to try to get 6 depths at each site, but on the more shallow sites, I collected four samples for each site. So here's a map made in ODV of the water column depth that you can kind of see. So here's north to south. So here are my northernmost sites, and those are the deepest sites. And I was able to collect 6 to 7 depths in the water column from those sites. And then as we got a little more shallow on the shelf, there were 4, I was able to get 4 depths. And I really targeted around the chlorophyll max and across an oxygen gradient for my samples. And these collection depths ranged from 2 meters to about 950 meters below sea level, so much deeper than my Gulf of Mexico sites, which I think the deepest site I ever sampled was 30 meters in the Gulf. So again, the depths are different from these two different ecosystems. My methodology was very similar to what I did for my dissertation work in the Gulf. Here I collected 12 samples from 12 stations across various depths and oxygen profiles. And I tried to do 10 liters at my deepest sites, but most of them due to time and the amount of water, 5 liters. I was most of the sites I got 5 liters of water. And then I also collected filtrate for nitrogen, and then dissolved organic nitrogen, dissolved organic phosphorus, and ammonia. And I stored all of these at minus 20 C on the research cruise. And here again are the Steravex filters where we collected the microbial biomass on these. And then in that green box are the samples with a bunch of ice to ship them home, get them home to the US to do the ITAG sequencing. But in the labs, so here's a map of the sites, the depths, and the oxygen concentrations for each. So back in the lab in Namibia, I used kits because I brought a lot of my tools with me, and kits were easier to transfer. So I used kits to extract and purify the DNA. And then I used the nanodrop provided in the lab to quantify it and amplify 16S RNA genes using PCR. And I also brought with me some AMOA primers. And this enzyme, this AMOA encodes the enzyme that's unique to nitrifying organisms, since I was really interested in looking to see if we could target Thaumarchiota. And then I visualized the results using the gel electrophoresis in the lab. So these are all tools that I was able to use in the lab back on shore. And what we saw, so this is looking at the phylum level again, is this Thaumarchiota that looks to be, and here we have suboxic, dysoxic, and oxic. So we broke up our data set into these three oxygen, I guess, characteristics. And you can see how this Thaumarchiota looks to be more abundant in the dysoxic area. So they're part of the top 12 most abundant organisms at the phylum level, and they represent up to 20% of their relative abundance in these dysoxic sites. And then the most abundant organism that we're seeing is a gamma proteobacteria, which represent over 60% relative abundance in our samples. And if we look closer at the top 7 phylum that we're interested in and look at them across this oxygen gradient, so here are box plots. We can see how here's the gamma proteobacteria, that their abundances look to be higher in suboxic waters. And then you can look at the alpha proteobacteria. So these are the next most abundant, and they are more present in oxic. Bacteroides they look to be everywhere. And then over here, if you see our Thaumarchiota, you can see that their abundances look to be higher in this dysoxic, the dysoxic part of the water column, as well as suboxic. And you can further look at the Euryarchaeota, which we saw in our Gulf of Mexico samples. They were also abundant and present in our samples there, look to have their niche in this dysoxic water column, as well as delta proteobacteria. So it really comes down to the electron acceptors, and that can help really define these metabolic niches and lead to different biogeochemical influences that these microorganisms have on the water column. And as oxygen minimum zones expand, they can impact these ecosystems and environments for these microbes. And so it's really interesting to study. And this is kind of just taking a quick look at the data set, and then Dr. Mason will then continue the story and take a closer look at the microbial community and what's going on in this environment. So I just want to say thank you for that this amazing experience that I had in 2017, and I think we really got a great data set from it. And it's starting to piece together these environments and what's going on with the microbial community in the water column of these oxygen minimum zones. So yeah, thank you very much. And I just want to thank the crew of the Mirabilis and Kurt and all the organizers of this course. It was really an amazing time and I learned a lot from it. So thank you. And
Thanks very much, Lauren.
yes.
Thanks for the great talk. And did you want to do some discussion now, or do you want to wait till after Olivia gives her presentation?
Olivia might help answer more questions, or she's going to go into a little more detail. But if there's, I don't have the chat up. If there's any,
nothing yet. Yeah, so maybe we can wait until.
Okay, but yeah, we could have an overall discussion of the whole data set after the second talk.